Alright, so good morning everyone and welcome back to my blog channel. So this morning, as scheduled, we will study about the genetic variations and Mendelian law inheritance. However, due to some tutor class was behind proceed before the schedule. So it leading to some class, some tutorial class, will have to take the exercise before the lecture. So in adjust, I put the chapter 6 related to the linkage list equilibrium and the population genetic first, so we can catch up with the exercise, and then we will come back with the other part later. Okay. So of course, in the beginning, I will tell you briefly about Mendelian inheritance, so we can catch up with the theory of Morgan and the definition of linkage inheritance. All right. So first, let's talk about Mendel. Mendel is considered as a father of genetics, and you know that when early childhood, the families Mendel was so poor that their parents cannot afford his expensive education. So Mendel was sent to a local chapter to study to become a priest. So his education will be paid by the church. So at young adult, Mendel was trained mathematics and physics. And when he completed his study in Trieste, he come back to his village to serve as a teacher in mathematics and physics. And then after a few years, he started his experiment in his small garden. And from this garden, he chose to study his subject in computer lab. And the funny thing is, Mendel had chosen so many physical traits to study in his career. But in court, we don't know that he chose the trait on purpose or just randomly, but his seventh trait that he to study in the fifth plant is located in different chromosomes. So in Mendel, he never had chance to study about genetic linkage. Now we have two hypotheses. First, he just randomly picking these traits, these 7 traits, which is randomly located in the different chromosomes. And the second hypothesis is he studies so many traits, and which traits is not following his law, he discusses it. Okay, so he just keep the trait that right with his theory. Okay, you know that Mendel is the first case of cheating in science that was discovered by Fischer. So story is, Mendel, during his year of science, he hired a gardener. Like nowadays we have a research assistant to help him to collect the data, collect the pea plants. And we don't know if he's doing it in purpose or not, but he tells also the gardener what is the expected outcome. For example, 3:1. We will expect the 3 ratio of the yellow pea plant and 1 green of the pea plant. So the gardener, to satisfy his boss and in order to keeping his job, throw away all the pea that not following the 3:1 ratio. Just keep the perfect calculation. And nearly a decade after, scientists in mathematics, Fischer, who invented the Fischer exact test famous, feeling that this result is too perfect to be true, even if the theory of Mendel was right. So he recalculated and see that even if the theory is true, the probability to obtain this is just still 0.0001%. That means we barely ever can obtain this perfect result in real life. Okay? Just like you throw a coin. Okay? You throw a coin 1,000 times, and the expected number is you will have 500 times in tails and 500 times in coins, right? So that is the perfect number. But actually, we barely, sometimes we can obtain this perfect result, but most of the time we will obtain something in between the perfect ratio, like we obtain 498 the tail, and 502 is head. Okay. So to obtain a perfect result, remember that sometimes perfect result is not good. Perfect result only obtained when we on calculation. In real life, there's always an adjustment. So, with Mendel, he has invented 3 laws. Law of dominance. That means when we cross true breeding parent, one with the yellow, with the trait of the color of the seed, yellow versus green, and because it's breeding, so we will have yellow is white, and small one. We will have the gamete. This one, BP is 100% white and 100% small. And in F1, the combination of 2 of these, we will have big one and small one. And the phenotype is 100% yellow So that is the first law of dominance. That when we have an individual that heterozygous with a genotype, then the carrier will carry the phenotype of the dominant allele. Let's call this dominant allele. And this is called the recessive allele. The law of segregation. So if we collect this F1 and make it auto-fertilization, auto-flaminate, we will have GF1 right now. 50% will be big Y and 50% big small y. Okay? And thus, this F2, we will have, let's take the table, 50% big Y, 50% small y. And here we will have also the 50% of big Y and 50% of small y. So that makes in here, the first combination is 25% big Y, 25% big Y, small y. 75% of big Y, small y, and 25% of small y, small y. Right? So, we will have genotype of the F2 is 1 or equivalent to 25% of big Y, 2 or equivalent to 50% of big Y, small y, and 1 or equivalent to 25% of small y. And in phenotype, so we have two big Y, one big Y is yellow, and two big Y, small y is also yellow. We will have 3 or equivalent to 75% of yellow versus 1 equivalent to 25% is green. That's it. And the last one is the law of independent assortment, which is if we cross more than 2 phenotypes on 2 phenotypes, from 2 phenotypes, because each phenotype, each paired allele will independently inherited. And when it's independently inherited, for example, we will have 3 to 1 cross with 3 to 1. So we will have, the ratio is 9, 3, 3, 1. Right? But the whole thing is, this third law is a weakness of Mendel. Okay? So at the moment, Mendel is just refer about the term 2 factor of inheritance, one from father and one from the mother. He don't know about the chromosome structure. He have no idea about genetic linkage. probability, he researched some other phenotype behind this, but it's not following his famous ratio 3:1, so he's discarded. And thus, he can never explain some other thing. And this law nowadays has been more explored, is only right. Third law, only right. Since the locus or the pair allele are located in different chromosomes. We'll talk about it now. So, talking about Morgan. Morgan was behind the Harvard Mendel just about from 20 to 30 years. He was the first native-born American who awarded with the Nobel Prize. And he considered us as the father of modern genetics. So when he doing his research with Whiteman and chose the pea plant, Morgan chose to study in fruit fly. The fruit fly have an advantage that it has a very short life cycle. Its life cycle is around 15 to 20 days. That means after 15 to 20 days, they will provide a new generation. So scientists don't have to wait long to see the result of the cross. Okay? And in order, the genome structure of the fruit fly is quite simple. Nowadays, fruit fly is still used as a host vector to perform many research that applies in humans. So, when Morgan performed his fruit fly, now at the time, nowadays we know about all the chromosome structure, about meiosis, about mitosis, about recombination after fertilization. But at that time, they don't know. So, Morgan performed the same experiment in fruit fly, which is an animal model, to see that if he can obtain the same result with Mendel when he applied in his pea plant. So he crossed two fruit fly, one with the yellow body and the other one with the black body. Okay? And these two fruit fly is true breeding. That means they are homozygous of the genotype to carry. They want to make sure of it. They have made it cross in the population so many times until the SOA phenotype is totally homogeneous in the offspring. So when he performed a cross between a yellow body and long wings with a black body and the short wings. In the F1, we have 100% is carry the yellow body and the long wings. So at this time, the experiment is still followed as Mendel inheritance, because the F1 is still showing one of the dominant phenotype, right? But the tricky question comes after. When Morgan take one of the F1 and cross back to one of the parent. The parent that carry the recessive phenotype. You take the F1 and cross back to one of the parent. So this is back cross. And in the result, he observed that in the population, we see in the population back cross, the result of this cross, instead of obtaining the ratio follow Mendel rule, he obtained 80% of the fruit fly showing the parental phenotype. So that means that 40% will have a yellow body and long wing, the same with this one. 40% will have the black body with short wing, similar to this one. And beside, he obtained 20% having a new phenotype, a recombinant phenotype. Yellow, black body with long wing, and yellow body with the short wing. This phenotype have never observed in the past. So now, at the time they know only about the theory of inheritance by Mendel. If Mendel were true, that means each phenotype were controlling by one independent pair allele. If Mendel were true, then we will have this gamete was equally distributed, right? So we will have 20% in this individual. We will have 25% will have this pair of chromosomes, big B, big S. 25% will have the big B, small s. 25% will have small b, big S. And 25% will have small b, small s. And when we cross this with the recessive phenotype individual, we will have the result as follows. 25% will have the yellow body with long wing here. 25% will have a black body with short wing here. 25% will have the black body with long wings. And 25% will have the yellow body with short wings. So, if the theory were true, this is the result we should observe. But instead, Morgan observed this. So, like I told you in the first section, biology and genetics is an experimental science. We provide hypothesis, we design experiment, we observe the result, and we draw conclusions. In here, the result shows something unexpected. That means there's something wrong with the theory, something that we don't know. So actually, Morgan is not the one who figured out about the chromosome. No. Actually, one of his students, Alfred student, one day he performed his experiment, replicate with the experiment of Morgan, and then he figured out everything. And in order to honor his professor, he had named the law of linkage trait following his professor's name, law of Morgan. So law of Morgan is actually not invented by Morgan, but invented by one of his students. Okay? So, in the future, if you invent anything, you are welcome to put your inventions follow my name. Okay? So, Alfred Stubben figured about the experiment of his professor, when the two fruit fly in here, we following the cross, but the unexpected result, different with Mendel, come because these two phenotypes is not independently inherited. So, the third law of Mendel was not applied truly on this result, because in here the two locus was located in the same chromosomes. So when it's located in the same chromosomes, during replications, we know that during meiosis, the chromosome was duplicate once, and then we will have two divisions to equally distributed the genomes into gametes, right? However, in meiosis, especially in metaphase 1 and metaphase 1, we will have a cross-link between two paired chromosomes here. And the percentage cross-link is depending on the genetic distance between the two markers. In this case, in the case of Morgan, the two alleles corresponding to the color of body and the length of the wings is 20%. That means 20% they are changed to happen the cross-link between these two positions. As a result, in this 20% case, we will have the part of the yellow chromosomes here with each chain with the blue ones. And as a result, we will have 20% of the recombinant chromosome here. And it's equally distributed, so 10% will have the big B and small s, 10% will have small b with the big S, and the other one, 80% is equally distributed. 40% will have big B, big S, and 40% will have a big B, small b, and small s. So with the other one, we have small b, small s, corresponding to 100%. And when we cross the two gametes of two parents, we will have the result like observed. 40% will have the big B, small b, big S, small s. We provide the yellow body and the long wing, because in this pair, the dominant allele will show up phenotype. 40% will have the small b, small s, so will show the recessive phenotypes. And 10% will have a big B, small s, will have the yellow body with the short wing. 10% will have a black body with the long wing. And the genetic distance, because Alfred had named, is follow his professor with a centimole. So 1 centimole is corresponding to 1% change. They are cross-linked between the two positions. Okay? Normally, the genetic distance is in parallel in the positive correlations with the physical distance. That means that if the two alleles, the further they are from each other, the more change that there will be a cross-link between the two. But it also depends on the gene regions, because some part of the genetic regions is less to have cross-link than the other. So the whole process is due based on cross-symbiosis. We have two positions in the chromosomes here, and if these are not high linkage, that means there are percentage to have crossing between those, then the percentage of crossing happen with corresponding to the genetic distance. Okay. Let's have an overview again to remind you about the meiosis process.
In this lesson, we'll explore the details of what happens. Meiosis, sometimes called reduction division, is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into two stages of cell division, called meiosis I and meiosis II. Meiosis I has four phases: prophase I, metaphase I, anaphase I, and telophase I. And meiosis II also has four phases: prophase II, metaphase II, anaphase II, and telophase II. Let's look at what happens during meiosis I. Prophase I starts with a diploid cell. Its chromatin contains two uncoiled, spread out sets of chromosomes, one from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of four sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase I, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase I, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase I, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase I with one chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis I ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes, consisting of paired sister chromatids. Both cells now enter the next stage, meiosis II. However, unlike meiosis I, DNA does not replicate before meiosis II begins. Once again, in prophase II, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During metaphase II, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they're called chromosomes. Finally, during telophase 2, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis II ends with 4 genetically different haploid daughter cells, each containing only one set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has 2 stages of cell division, called meiosis I and meiosis II. During meiosis I, homologous chromosomes separate to produce 2 haploid cells, each containing chromosomes in the form of paired sister chromatids. In meiosis II, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produces 4 genetically different haploid gametes. And here are some key points to remember about prophase 1. The pairing of homologous chromosomes, called synapsis, occurs. Each pair of homologous chromosomes, consisting of 4 chromatids, is called a tetrad. During the process of crossing over, chromosomes in homologous pairs exchange segments of alleles. Crossing over results in genetic differences in gametes. All gametes produced by meiosis are haploid.
So, in the genetic listing, 1 centimogon is equivalent to 1% of the crossing between the 2 pair of the locus, right? So, as we can consider, that means if there are 2 alleles, even they are located in the same chromosomes, are independently inherited. That means that the genetic distance between 2 random paired locus can never excess 50 centimogon, which is equivalent to 50% of cross length between the 2, right? And in case that there are 50% of crossing, that means they are independently inherited. However, so first, let's have a small aside to remind you about the crossing between 2. So in here, I have the back cross, like what Morgan and Donovan have performed. And please calculate for me the parental phenotype and the recombinant phenotype if I have the genetic distance between the 2 is 30 centimogon and 0 centimogon. Please. Just a very simple exercise to idea about Markov law and linkage disability. Anyone have answer? Very easy one. Very easy questions. Yeah. Sorry? No. Sorry. So the first case is the genetic distance between B and S is 30 centimeters. That means there's 30% of cross-link between B and S. And the second one is, case is 0 centimeter. That means that there's no cross-link between B and S. Or in other words, in the second case, they are in full linkage. Okay. So, to give me the result. The second case. Yeah. So the A question is calculate if the Z is 37 and B calculate the proportion if the distance is very the distance between is only and yeah, please. Okay, that's correct. So actually it's a very easy issue. So in here we have genetic distance between B and S. The first case is 30 centimeters. That means there are 30%. So we have the big B and corresponding to big S and small b and corresponding small s in here and two are the same. So the first genetic distance is 30 centimeters. That means 30% of cross-link between B and S. Alright? So we will have the 30% of crossing here. That makes the gamete will be the big B and small s will be 50%, and the small b, big S is 50%. So the two recombinant is 30%, equivalent to 30 centimole of exchange. And of course, because this one is 15 and 50, the other will be 35 and 35%. Big B, small s, small b, sorry, big B, big S, and small b, small s. Okay? So the other takes 70%. Okay? Now, if we cross big B, small b, and small s, 100%, so we will have the F1 equivalent 35%. 35% we have yellow body with long wings, 35% we have black body with short wings, so it is 35% here. And the other 50% we have black body with long wings, black body with the long wing, and 50% we have yellow body with a short wing. Okay? And in other case, if the genetic distance is 0 centimole, then we have in the second case 0 centimole equivalent to 0 cross-link, so that means that we don't have the recombinant chromosome, 0%. And we have all of the gamete will be big B, big S, and small b, small s. So 50% of this. There's no cross-link between the two. So the F1 will be 50% will have yellow body with long wing, and 50% will have black body with short wing. And we have no recombinant here. Okay? Now, from genetic linkage to gene marking. Let's say that we have 3 pairs of genes, A, B, and C. Okay? Now I have the genetic recombinance between A and B by cross is 25%. I have the genetic distance between A and C is 13%. And I perform the genetic cross-link between B and C is 19%. Could you help me to identify whether the order of gene is A, B, C with the B is in the middle AC, or BAC with the A in the middle of BC, and the C in the middle of AB? Please. No. It's just the distance between A and C is 30 centimole. Like the distance between anode and hydrogen is equal to distance between hydrogen and hydrogen. Yeah, the plus. We don't have the negative distance, negative value in the genetic distance. We never have. Bạn hiểu câu hỏi nhỉ? Ok. crosslink crosslink là đã học từ cấp 3 rồi đúng không? Này học cấp 3 chả là đỡ gì. Nhưng bây giờ tôi có một bài toán là bây giờ tôi có 3 cặp gen A, B, C. A lớn A nhỏ, B lớn B nhỏ, C lớn C nhỏ. Tôi thực hiện phép line thì tôi thấy là cái tần số xuất hiện crosslink, tần số xuất hiện hoán vị gen giữa A và B là 25%, có nghĩa là khoảng cách di chuyển giữa A và B là 25 cm. Chưa? Thực hiện phép line giữa A và C tôi thấy là khoảng cách di chuyển là 13 cm. Thực hiện phép line giữa B và C thì khoảng cách di chuyển là 19 cm. Thế thì bây giờ cả ba gen A, B, C này cùng nằm trên một nhiễm sắc thể. Tuy nhiên tôi muốn xác định là bây giờ thứ tự gen sẽ là A nằm giữa B và C, A sang đến B sang đến C, hay là B sang đến A đến C, hay là C nằm giữa A, B nằm hai bên. Dựa vào cái phép này ABC, hai sinh viên chọn khoảng cách là ABC.
ACB đâu trở thành ACB BC BCA.
Rồi những ai chọn ACB giơ tay.
Em ấy mưu sinh bây giờ trong vui
5, 6, 7, 8, 9. Rồi khoảng độ 20%. Những ai chọn ABC giơ tay. 1, 2, 3, dưới 5%. Rồi ai chọn BAC giơ tay.
chỗ còn lại.
Alright. So to identify this, let's begin with AB first. Okay. So distance between A and B is 25 cm. I draw a chromosome, and I take a fictionally distance between A and B is 25 cm. Okay. Now, C could be located in here. If C located here, we have the distance is BAC. If C is located in the middle, we will have ACP. Distance between A and C is 30 cm here. And distance between A, so it could be here or here. With the genetic distance is 30 cm. However, if we take the distance between B and C is 19 cm. So if C is located in here, then the distance between B and C must be more than 25. Okay? It must be 30 or something. But in fact, we have the distance between B and C is 19. So we must have the C is located in the middle of A and B. So the true order is A, C, and B. One more thing to remember. In gene mapping, in the genetic distance, we don't have the additive rule. That means if we know that C is located between A and B, we cannot take the distance AB is equivalent to AC plus BC. Because as we can see here, the distance between A and C is 13 cm. This distance between B and C is 19 cm. So the total distance is, let's say, 32 cm. But in fact, we have only 25 cm. So there is no additive rule in genetic distance. We might have the additive rule in the physical distance, but we don't have the additive rule in genetic distance. I'll take example. The genetic distance and the physical distance. So in the physical distance, if I have the distance between A and B is between A, B, and C. If I have the physical distance between A and B is 3 kilobase, I have the distance between B and C is 7 kilobase. That means B and C are far away from each other 7,000 nucleotides. I will have the distance between A and C is 3 plus 7 equivalent to 10 kb. Okay, so this is a additive rule. Okay, but in genetic distance, if I have A and B is 10 centimeters, between B and C is 7 centimeters. We don't know distance A and C with less than 7 plus 10 plus 7 equal to 70 centimeters. Or in other words, no additive rule. There is no additive rule in genetic distance. Now, another exercise. I have the genetic distance between A, B, C, and D as the following table. Please draw a map of these 4 G forms. Anyone have solutions? Yeah.
Man says B, A, D, and C.
B, A, D, and C. Okay. Others? You have the same result?
Yes.
Great. So the correct answer is B, A, D, and C. What should we say? It's pretty easy. We have BC is the longest one with 35%, and the A is A in the D must be in the middle. But BA is 20%. Meanwhile, B and C is 35%. So B must locate near to A than C. A here. The BD is 35% and the AB is 20%. So B is located to A nearer than D. So we have the order is B, A, D, and C. And that's the same when scientists in the 19th century want to identify the genetic mapping, because at those days they don't have the sequencing technology. They don't have the NGS, the next generation sequencing. So all they know about the genome is just through a cross between different individuals in the species. So in here is a mapping of a fruit fly based on a phenotype. So we have the distance between a short arid state with the marine eyes in here is 16.5 centimeters. That means there is a cross-link between the percentage of having cross-link between this 16.5%. Okay? We will have the distance between short average state black body is 48.5%. But then we will have something strange. The distance between short average state and cinnabar eyes is 57.5%. So what does that mean? Because we just discussed a few minutes later that the frequency of crosslink can never exceed 50 centimoters, right? In case 50 centimoters, that means it independently inheritance. But why in gene mapping we have this one is 57.5%? Is there something wrong? We have the genetic distance in here is 67 centimorgan, and the last one in here even 104.5. That means that the crosslink between this one and this one is even more than 100%. Is there any explanation for this? Any explanation? So, actually, that's a way to draw a genetic distance. So, the distance in here is 16.5 centimorgan. That means 16.5% that is cross-linked between the two here. This is true. But if we compare this one, the black body, the colored body, and the length of the arid state, so the genetic distance in here is 48.5%. Doesn't mean that there is 48.5% cross-link between these two. So the centimorgan in cross-link is different with the centimorgan in genetic distance. Now, we refer to a different genetic map. A and C. B in the middle. We have something on the genetic distance in here is 10 centimeters. Okay. We have the genetic distance in here is 15 centimeters. So in order to draw a genetic map, they will take this as a pinpoint. So if the genetic map is starting with A, then the distance between A and B is 10 centimeters short, equivalent to 10% cross-link. But if we draw a genetic map, I'm saying we take AC and we say that this one is 25 centimeters. And we have the middle is B. So that means that the distance between B and C is 50 centimeters, equivalent to 15% of cross-link. And this one is different with 25% of cross-link. The same with this one. So if we say that in this mapping, the distance between black body and star RXX is 48.5 centimeters, doesn't mean that there is 48.5% cross-link between body and the length of RXX. It just means that the cross-link between black body and minor eye is 32%. Okay? Because the 48 minus 16.5 is 32. So you understand this point, right? Not really. Okay. Cái giải thích lại thì thực hiện. Trong trao đổi chéo trong hoán vị gen thì tất cả tiểu học thì các bạn rồi. Tần số hoán vị gen không bao giờ quá 50 cm/g, đúng không? Tương ứng với 50% trao đổi chéo. Và trong trường hợp 50% trao đổi chéo thì đấy là phân ly độc lập. Phân ly độc lập thì tần số cũng là 50%. Tuy nhiên khi đề cập đến việc là xây dựng cái bản đồ di truyền thì người ta không quy ước một cách tương đối, không quy ước một cách tuyệt đối như vậy. Ví dụ như trong bản đồ này, đây là một cái bản đồ di truyền mà các nhà khoa học khi mà chưa có công nghệ giải trình tự, không có công nghệ đọc trình tự gen thì người ta làm trên cách như thế này, là người ta lai giữa các thế hệ với nhau để xác định xem là khoảng cách giữa cái cặp tính trạng nào so với cặp tính trạng nào là gần nhất, xa nhất. Thì ở đây ví dụ như giữa hai cặp tính trạng liên tiếp, ví dụ như là short Aristotle là vô dài và tính trạng màu mắt là khoảng cách di chuyển là 16,5 cm/g. Có nghĩa là ở đây cái tần số trao đổi chéo giữa hai cặp tính trạng này là 16,5 cm/g. Tuy nhiên sau đó người ta thấy giữa hai cặp tính trạng này cái khoảng cách di chuyển là 32 cm/g, tương ứng với 32% trao đổi chéo. Và khi người ta vẽ bản đồ di chuyển thì nếu người ta đã xác định cái điểm này là mốc thì người ta sẽ phải cộng một đoạn 32 cm/g nữa đây, dẫn đến việc là cái khoảng cách di chuyển giữa cặp màu thân và cặp độ dài râu này là 48,5%. Điều này không có nghĩa, tôi nhắc lại, điều này không có nghĩa là tần số trao đổi chéo giữa black body, gray body và long Aristotle là 48,5%. Mà nó chỉ có ý nghĩa giữa hai cặp tính trạng liên tiếp, đúng không ạ? Ví dụ ở đây giữa hai tính trạng này tần số trao đổi chéo là 9%. Điều này không có nghĩa là tần số trao đổi chéo giữa cái này và cái này là 57,5%, hay giữa cái này và cái này là 48,5%. Nó chỉ có ý nghĩa giữa hai cặp liên tiếp. Chưa? Hiểu chỗ này không? Đúng rồi. Ví dụ như là cặp này, tần số trao đổi chéo sẽ là 29, 100%. Nhưng nếu mà mình lấy cái này là khoảng cách di chuyển giữa cái này là 104,5%. Không có trao đổi chéo nào quá 50% cả. 57% đã là 57,5% đã là vô lý rồi, đúng không? Cái này chỉ có ý nghĩa là khoảng cách giữa cái này đến cái này nó còn thông qua rất nhiều cảm khác nữa. Và đương nhiên là khi mà mình có những cái marker mới, tính trạng mới insert ở đây thì cái số cái khoảng cách di chuyển giữa cái những chất thể này nó sẽ tăng lên. Được không? Okay, so the same when we building the genetic map between in the potato plant. So in tomato plant, we will have 12 paired chromosomes, and this is the genetic distance in each paired chromosome. So, for example, the genetic distance in the whole chromosome 1 is equal to 12 plus 4 plus 17 plus 16 genetic distance of chromosome 1 is equivalent to 12 plus 4 plus 17 plus 16 5, 4, 6, 4. Which is equivalent to, can anyone make a calculation for me?
Dưới cái con số đó, tự nhiên 78 ạ. 78.
78 centimeters. So that means here we have the genetic size of the chromosome 1 is 78 centimeters. And remember that the cross-link was affected by gender. Okay? Like in fruit fly, we have the cross-link was affected by gender. And also in human, we will have genetic map were affected by gender. For example, here I have chromosome 12. And the marker here in the woman is different with in man. So in man, the genetic distance between these two is 23, meanwhile this one is 10. But the genetic of the whole distance in woman is larger. That means what? That means in chromosome 12, we have this structure of chromosome was more unstable in female compared to in male. Okay. Alright, so now we will have a short break, 5-minute break before we go in proceed in the next chapter. 5-minute break. Lại lại một tí nữa đây. Ở đây mình có là tất cả những cái marker của cái nhiễm sắc thể số 12 ở người. Tuy nhiên mình có thể thấy là tổng khoảng cách di chuyển ở nữ là nhiều hơn ở nam. Điều đấy cho thấy là gì? Giữa hai điểm này chẳng hạn, tần số trao đổi chéo ở nữ là nhiều hơn ở nam. Như vậy khi mà tổng khoảng cách di chuyển của nhiễm sắc thể lớn hơn ở nữ so với ở nam, có nghĩa là cái nhiễm sắc thể này ở nữ nó sẽ không ổn định bằng ở nam. là tần số trao đổi chéo của những sắc thể này ở giới nữ sẽ cao hơn ở giới nam.
ABCD này ngày xưa ông cứ học cái phương trình ấy, ví dụ như cho A cộng B bằng 35, B cộng C bằng 60. Và ông giải thể hình ABC. Ngày xưa cháu đi học ngoài học đấy đây áo học xuống đấy rồi dưới kia đây đây trên là nó khổ lại là sinh viên nghèo đã bắt buộc. Còn một cái nữa là ông có hiểu cái là lúc nãy thầy có nói là quy định phân ly độc lập lai tình trạng ấy là không được quá 50 quá 50% là phân ly độc lập. Lai tình trạng còn cái bọn kia nếu thầy sao thầy kiểu 56, 74 rồi chẳng mình ở kia ấy bọn này là bọn họ phân ly độc lập, nhưng mà với cái bọn lần trước họ ví dụ con 4 với cả con 74 thì con này nó là lai tình trạng. Vì sao? Nếu con lấy 44 xuống 74 đi nó sao thì là mình chẳng có. Rồi con này phải để về là cái ông kia ông có phải like kiểu đi từ như này đó, không phải là ông đi sang con này like dần dần sau đó họ đi sang này được like này thôi dần quen.
Mắm mù.
Vừa nói xong, con mạch là dài nhất. Còn A C dài nhất đúng không? Hai đầu đúng không? Em nói đi. Tôi nói khi nào rồi, tôi biết trên bảng này là ở trong sinh học, cái line là không giống như là cái vật lý đâu. Tức là ví dụ con này đây, hai ở chỗ này hay 200 đúng không? To dưới to từ nào bên vào bên ngoài.
thấy chưa đầu tiên là đây hôm qua mẹ ai đâu cái hôm nay đánh ấy anh Minh Dương rồi tao chơi dệt nhá, không có đi với thằng Mê ông nữa, thế là tao phải thằng Mê ông entry đúng không? Mẹ anh Minh còn entry cũng nhanh hơn cả tao phải Mê ông, thế còn cầm bom nó cầm switch cầm bom nó tự
10 tiếng đầu.
tiếng nào tao đã bật A thì tao định dash mà tao thấy thằng nào phía trước còn Minh tao nó chết nên là thôi kiểu tao cầm kiểu judge ấy, tao định kiểu lướt vào người lướt gần người team địch để tao đục, ai ngờ nó chết tao phải lướt là tao dash là tao nhặt bom hết này lướt cay vãi đái à. con dệt anh entry vào nó cũng có cái dash để nó khỏe hơn rồi.
Có một đấy, tao có con fit đủ stick mấy hệ rồi, tao vứt ra ngoài tao chắc là nó vẫn thua, còn câu hai. Tôi có fit đủ.
Cái con gì mà tao xem một cái con Syndra vứt mấy con ở ngoài con chip.
Con đấy là một trong những điều tham vàng.
Được cái vứt mấy con ở ngoài con chip. Tao chơi từ đéo lắm, tao chơi từ cái mùa còn cái gì ạ?
Ngoài con chơi ngăn chặn nó.
Đùa lít đấy, nhớ đùa lít rồi.
Right, quiet please.
Xong đấu ấy.
Bây giờ mùa này con xong đó.
Quiet please.
Mùa Zone đấy á. Zone ra xua.
So I will spend a few minutes of today's session to reintroduce to you the basic principle of population genetics. Quiet please. Quiet please. So modern population genetics is a science that study about the genetic flow stream of a population. But it's based on the classical population genetics with following the uneven equilibrium. Quiet please. Quiet please. So on today's duration, there is not much of time, so I can only introduce you the foundation of population genetics, which is the uneven equilibrium law. I cannot go further detail on the calculation, the population genetic flow stream. If you want to study such matter, you can following the further course in years, in second year and in third year of the PMAP department, in LS department. Okay? So Weinberg is invented by two scientists. The Artie, which is the English mathematician, and Dr. Weinberg, a physician. So the whole law is based on fireplace. The whole law is based on background that genetic distribution of the population will not change through generation. That means that the structure of the genetic populations was the same in different populations. So if we have genetic populations, the distribution will be equivalent to, for example, if the distribution of the gene with two alleles in the population is p plus q squared is p squared plus 2pq plus q squared. And for t allele, we will have this form. So what is p and q? So for example, in the populations, we will have the p and q is allele frequency of a two allele locus. For example, if a gene has two alleles, big A and small a, then the p is equivalent to the frequency of big A, and the q is equivalent to the frequency of the small. We will have the frequency of the genotype big A. If we have populations, so if we have the gene with two alleles, big A and small a, we will have the P is the frequency of the big A and the Q is the frequency of the small a. Of course, because we have only two alleles, so frequency of A and small a is equivalent to 100%, right? Because in the populations, we have only two alleles, A and small. So now what is the frequency of the genotype big A? So in populations, if we have the frequency of the big A is A, so then the genotype big A, for example, this one is 10%. Then the percentage of big A is p squared. If it is randomly, we will have p squared, which is 10% squared, which is 1%. So the first one is a condition, and the second one is the Artin-Weinberg law. If the population following the Artin-Weinberg law, we will have the frequency of the genotype big A is p squared. Otherwise, if the populations follow Artin-Weinberg theory, case not.
con này nó dính đùi cho con sinh con nữa con làm tiếng bộ này.
Then the frequency of big A, big A. Then you will have the frequency of A is equal to the frequency of big A plus the frequency of big A, small a divided by 2. And the same frequency of small a is the frequency of the small a plus the frequency of big A, small a divided by 2. Right? Because each individual have a heterozygous big A, small a will carry a half of the frequency big A and a half of frequency small a. So in case now the frequency of big A is equivalent to big A plus frequency of big A and small a divided by 2. And we got this in your bioinformatics course, right? For example, go on Ansible. So in your bioinformatics course, we will have questions related to the frequencies. This one.
Mua vào cho nên chẳng thích nữa.
Go. And let's see that the genetic distribution of this one in different populations. And you can check also for the other marker. The internet connection is really bad. It takes too long. So, it doesn't sew up. Alright, fine. Click on genetic populations. Population. It's really crazy internet connection. Alright, we will wait it a little while. So in here, let's come for an example. We have populations that in only one band equilibrium. That means that the genetic distribution of each population are the same in different populations. So, if a population have a 36% of the population homozygous with a recessive allele A, what is the frequency of the recessive allele? answer? We have populations may not be one magically mean. We have a frequency of small a is 36%. So what is the frequency of the recessive allele? We will have the frequency of the recessive allele in here is square root of 0.6, which is equivalent to 0.6, corresponding to 60%. Remember that this population is optimal. What is the frequency of dominant allele A, big A? So from this we will have frequency of big A is 1 minus 60% is 40%. What is the percentage of what is the frequency of the big A, which is equal to the frequency of big A squared of 4 squared 4.60, okay, which is equivalent to 60%. And the frequency of big A small a is equivalent to F big A and F small a is equivalent to 0.4 times 0.6, which is equivalent to 48%. So we will have a population with 36% of small a, 48% big A, small a, and 60% of big A. So the RD1 map is outlined for the population that is stable of the genetic structure. Let's hypothesis that this population undergo auto multiplication. And this one is totally random. So what is the frequency of the small a? In this population? Small a is 0.6 is 60%, right? Big A is 40%. Right? So we will have, if this one is undergo fertilization, we will have 60% of small a, 40% of big A. 60% small a and 40% of big A. We will have the genotype small a is what is the percentage of small a? Again, the frequency of small a is equal 60% times 60%, right? Which is square, which is 36%. And what is the percentage of big A, big A?
Ta lên béo người béo còn đi ta chụp lưỡi
0.4. 40% times 40%, which is
như kiểu tổng lý khổng chết chứ
16%.
đúng hai đục
And what is the percentage of big A, small a?
operator I don't think
So it's
muscle
equal to 40% times 60%, which is 0.4 times 0.6 is 0.24, and in here again 0.4 times 0.6 is 0.4. So the big A, small a is 2 times 0.4 and 0.6, which is 48%. So again, if we have this population was undergo auto-fertilization, we will have 36% of small a, 16% of big A, and 48% of big A, small a. The same with the previous generation.
lấy lấy đi vậy
So that means that this population are equivalent to optimal equilibrium. Okay?
chú chú hai mấy
So let's go on reality.
chiều
chiều theo mấy dịch cắm người đi nhé
cho chú đập ấy
ở đây này
chú đập
So of course, if we want populations to undergo the stable in the population in here, we will have a certain condition that this population will have to maintain the optimal band equilibrium. First, we will have this population must not have mutation. Genetic mutation can change Allen frequency, okay? And Allen frequency change can change the genotype distribution in the population. We must not calculate gene flow or migration. For example, we must have the population is quite isolated. A migration, a flowing genetic tree will change the whole genetic population in the genetic distribution in the population and make the stable condition was great. Non-random mating and natural selection. Okay? Natural selection can make the frequency of one of the two alleles change in the population. For example, if this small a was affected to the vitality of the carrier, that means this flow gene stream will not passing through the later generation, keeping the break the stability of the genetic distribution. And finally, the population must last enough to avoid the genetic drift. The genetic drift is, for example, like the distributions, the eliminate of a certain genetic bloodstream out of the pedigree because of the vitality of the character. For example, if we have you have all go through the maneuver.
Bàn tay.
So, for example, if we have populations in here that both parents are not carrying disease, but two of the offspring carrying disease, and these two was eliminated by birth, or before they can have an ability to produce baby. That means this allele will be eliminated out of the population. Get into the genetic drift. The random trait that this one will be eliminated out. Okay? Not because of the survival of the allele, not because of the natural selection or free-phase mating, but randomly just by luck. Okay? Alright, I think today session will be finished here. Do you have any questions? Okay, since you have no questions, I think today's session will be finished here. Thank you very much and see you next week.
Bên Raven tao đánh trận nào cũng được kiếm làm trắng.
Có bóng rổ là nhìn đẹp mã của chị cầu thủ cấp đội cái bóng rách dưới không có cái bóng tan nó cũng Let's go.
Có Raven phải đầu game thì phải xoay có vài cái.
Còn đấy vị trí